Hi everyone, thanks so much for joining. We have a lot of people. It's probably the most popular trial ever launched in SeedLinked, so it's crazy. Thanks so much, Sarah, curator of Bean, who is willing to be the guinea pig for the USDA seed bank. But I'm Nicolas Enjalbert. I'm based in Viroqua, Wisconsin, west of Madison, co-founder of SeedLinked. I've been a breeder for a while and leading SeedLinked in many different directions, from breeding to characterization. And here today, we are really more Q&A. We're going to give some intro of the trial. Sarah's going to present the collection. I'm going to give a tour of SeedLinked just to go over a few things and help you. And I know you already have a lot of questions, so we really want to have at least half of the time for questions. But let's start with a quick intro. So that's from me. Alice is,
Okay.
go ahead.
Yeah, hi, I'm Alice from eOrganic at Oregon State University, and I'm just going to be in the background running the webinar, and I'll moderate the questions afterwards. And I just wanted to let everyone know that the recording will be available within a week after this webinar is over, and Nico will put it up on the SeedLink YouTube channel. Is that right, Nico? And link it to
Yep.
the SeedLink site? Okay.
Yeah, and in the feed as well, so you can rewatch, share it. So it will be posted in multiple channels.
Great. And I'm Sarah Doley. I'm the curator for the USDA bean collection. I have a passion for growing and eating beans. I used to teach, I used to be a bean breeder, and I'm so excited about this pilot project and the volunteer enthusiasm.
Yeah, it was such a great project, and with bean literally from all over the world. I'm just going to give a quick intro of the trial, give you a few numbers, and give the hand to Sarah. I'm going to share my screen quickly. Yeah, in the agenda, just a quick description, then pass to Sarah, then I'm going to take over, and then I hope to have at least 30 minutes of Q&A. But just a few numbers before Sarah going to describe the collection. We we originally were planning to test 10 to 15 accession as a pilot. We were looking for 100 people as a high number, and we launched the trial. And in a few hours, we opened the registration. We had 260 people sign up, so just never happened before. We didn't have enough seed. We succeeded to find more seed, and we ended up now to have 213 growers. We had to turn down people across 44 states. So it's such an amazing coverage of the whole of the US to test those bean, and there's 21 accession total, 20 from the collection, and one extra check from Europe. I succeeded to find to add 10 additional people. And you all receive a random combination of those 21. That's the model we use to get the most accurate data. So each variety was submitted and is going to be planted about 30 times across the US. And you all receive 7+ seed per variety. And yeah, you're really all encouraged to plant them together. Really make sure to well label them. We put it stakes in the package, and so make sure that you write their name with UV proof, waterproof label to track them. And yeah, so that's kind of the distribution of all of you part of the trial. And so it's really good. We have a really good representation of the US. The Central West is all the time less people, but it's the mountains. But and yeah, from here, I'm gonna let Sarah take over and talk about the collection, the need, tips. I know because some of those variety are unknown, or you're really here to bring information. And so
Great.
here we go.
Thank you, Nico. Thanks for the intro. Can you see my screen now, the PDF?
Yeah.
All right.
Yeah.
Welcome everyone. I am so excited, and everyone here at my unit is so excited. So my job is to grow and characterize beans, and to think that there are 200 plus volunteers who are interested in helping me is just so validating. So very briefly, I'm going to give you a background of the entire national plant germplasm system, which I'm only a small part of, a background on the Phaseolus collection that you're hoping to characterize this summer, and a little bit about the plan for this summer, with a big reminder that this is a pilot practice year. So we have a lot to learn on our end, and you'll have some to learn from your end. And if it goes well, we hope to do this on a much larger scale in the future. All right, so the National Plant Germplasm System is made up of 20 locations across the US. And they're different gene banks, right? They're not just seed banks, because we also store clonal things like trees. Here they are, the different locations set on the growing zone map. We also collect crop wild relatives for conservation, as well as wild crops. We have over 600,000 accessions conserved, over 16,000 different species. And in addition to the conservation, we also do utilization. So we share these resources freely with public and private breeders and researchers all around the world. And we have longstanding partnerships with universities and ag extension stations. So the bean collection, which is held in Pullman, Washington, Eastern Washington on the border of Idaho, contains over 17,000 accessions of Phaseolus beans. Those are the New World beans. So your common beans, which you'll be looking at this summer, lima beans, tepary beans, runner beans, Demosis, and then 51 other wild relatives. 75% of the collection is available, which means we have more than 300 seeds of each of those, right? Almost 75% of it is backed up. To be backed up, it's in long-term storage in Fort Collins, or we have further backup at the Doomsday Vault in Norway. We spend a lot of time here doing seed increases. We grow all of the beans in greenhouses every year to keep them free of bean common mosaic virus. And then we on average grow about 330 accessions. And each year, something we're really proud of is the distribution. So we've been distributing over 5,000 seed packets a year. And we're distributing not to home growers or community gardens, though we love those. We're distributing to researchers and breeders, because we don't have varieties that are necessarily going to thrive in someone's garden. We have some seeds that are over 20 years old that have low vigor. We have some seeds that are photoperiod sensitive. We have some seeds that don't taste good, right? And they're not ones that home gardeners generally want, but they might contain some genetics that a breeder can put in to make an improved variety, right? We also work on new acquisitions. So just in this picture here, we have a Demosis, white flower on the left, and a lima bean flower on the right. They're two different species, and they were mislabeled. So that what we're doing is verifying the species, and we're growing some stuff outside, and we do a lot of it in the greenhouse. So our regens, which take up a lot of the time, are all done in the greenhouse. So challenges of keeping the vines separate from each other, right? And then just greenhouse space. We can only plant 380 accessions at a time. Some of them take more than a year to produce seeds. If they're perennials, a lot of them are photoperiod sensitive, so they won't grow here in the summertime. Just a quick graphic on our seed distribution. You can see over time it generally is increased. And the breakdown, I think is fascinating. The blue section is the vulgaris, which is our common beans, but we get a lot of distributions of our lima beans, our tepary beans, and our more minor species, which shows that this genetic diversity we have in our collection is of value to the community. All right, so characterizations, and this is where all of you are going to come in. Characterizing is phenotyping the collection. So when does it flower? How does it taste? Is it disease resistant? And that is something that's really hard for us to do here in Eastern Washington, growing our beans in the greenhouse, because people don't grow beans in greenhouses. People grow beans in gardens and in fields. So we need you to grow them in your garden in all of your different climate conditions to tell us how they're growing, right? And just a note here, you can see the picture in the top right has a straw-colored pod. When we're talking about seed maturity, most of the pods will be straw-colored. At least they'll be dry and the seeds will shake a little bit. Okay, so the traits we're looking at would be, and this is recorded so you can look at this later, and the information is going to be available on SeedLinked. But your planting date, if it's photoperiod sensitive, the growth habit, if it's an early bean, people like that. And then stuff that's interesting to you, which is how easy it is to grow and how good it tastes when you cook it. So growth habit is something that's really important, and we're figuring out how you are going to report this in the SeedLinked app. But this is really important in the bean world. We have bush type beans and we have vine type beans, right? And a bush type bean, the way you know it's a bush, it's not necessarily the height. It's because the terminal meristem or the apical meristem, the shoot tip, switches to a flower, right? And that would become a bush. That means there is no more vegetative growth when it's flowering. And then the pole or the vine type, those will all continue on their terminal stem to be vegetative. So if you can't tell, take a picture of it and we'll help you tell. And I'm sure there'll be more questions about that. So that's a bad cartoon, but this is zooming in. So this is a challenge in the greenhouse because if you force a bean to climb, a lot of them will climb. So this is actually a bush type lima bean that's over 6 feet tall because we made it climb, right? We restricted the light. But looking at the tip, the terminal meristem, the shoot tip, zoomed in, you can see it's pods and flowers at the end. It's no longer making stems and leaves. So this, even though it's 6 feet tall, is actually a bush. And that's really helpful if you can tell the difference. If they grow in the field, which is what you'll be doing, they're going to look different. We have climbing vines, we have prostrate or sprawling vines, and then we have bush, right? And that looks like a bush when it's outside because we're not forcing it to climb. Right, so here at the MPGS, we're trying our best to maintain the super diverse collection, and we are really looking forward to you helping us characterize it. And we actually put a strategic plan forward saying we're going to characterize the whole collection. So depending on how this summer trial goes, we'll modify it and then in the future characterize some more together. This is my contact information. It's my first name, sarah.doley@usda.gov. If you want to email me. And here's the link to the strategic plan if you want to learn more about the entire gene bank system and what we're up to.
Great. Thanks so much for the intro, Sarah. And please, try to use the feed in SeedLinked, and I'm going to give you now kind of a tour of the platform, but to try to keep the communication under the trial feed. I'm sure Sarah is totally swamped with work, but I'm sure she would love to respond to questions, but
I think that's a great idea to put it through the SeedLinked feed so that we can show that this platform is a good one. Because there's a lot of other curators who are interested in utilizing crowdsourcing for genotyping, but we don't have capacity to do any more work. So anything we can do to make it less work for the curators, they'll be more in favor of sharing their collection for this.
Yeah, I mean, really this pilot all together here with all of you is to showcase a way that will give efficiency and capacity to our seed bank. I mean, what Sarah's doing is a public service, and it's all this germplasm. And how can we give more capacity to give this information that will all help us and scientists and bring novel line and so forth. So anyway, amazing. I get so excited of all those things. I'm gonna start sharing now the SeedLinked just to give you to kind of bridge a little bit what Sarah said and how to use SeedLinked. And also, as usual, and we will do a survey at the end, but you can also send feedback to SeedLinked all the time on how to improve it. We love that, as you know. Yeah, and just starting a little quick overview for some who don't know as much what is SeedLinked, but the principle is here we are connecting hundreds of growers, but imagine the whole network of SeedLinked is tens of thousands of growers. There's 32 million households who grow vegetable in the US. Imagine connecting even just 1% of those, and what vast amount of knowledge we could gather to not only help any other grower to find another variety, but help people like Sarah, scientists, breeder, what could be the power. And that's the foundation of SeedLinked, to connect this information to have really a multiple purpose of action. And the platform has really 3 big pillars. It has really this collaborative R&D where it can be part of a trial, record information, but there's also a peer-to-peer communication, exchange, a feed, because collaborative innovation go hand in hand with communication and interaction. So we have a full social media feed within each project, also across, so you can follow each other. And then we have a search engine that really connect all the dots and all the information and make this information available for search for you to find information. And we have already, after 5 years, more than 500,000 information from insight from grower. And so what you can do at SeedLinked as a grower, well, you can join a trial like this bean trial. You can also track you can create your own trial in the app and track your own garden and share information of what you grow. You can obviously exchange in the feed, interact, ask questions, build your own community, and search and find new variety. And so from here, just going to dive into the bean trial and really focus on trialing. I highly recommend you to download and use the app. The app can be used without connection if you put on airplane mode, but you don't have to. We also have the web. And I know some of you love going in the field with a clipboard and a paper and take note and then go back home and enter this on the web, and you can do that. But really the app is really user-friendly and getting better and better every day. And so I will just give you a quick tour of the app. I think, is this on the, okay. You can see on top when you're on the track is where all the trials are happening. And each tab is a trial here. And so you may have only one trial, you may have multiple trials if you're part of some adapt trial and so forth. And when you click on it, I will show you later, but at the bottom, you can access the feed, the search, your profile and notification. And so when you click, you can see each trial has 3 little dots here who give general information. You can record your trial management log. How was the management of your trial? You can see the description of the trial that we set with Sarah, who gives the main goal of the trial, few instructions, and same, they're accessible there. You can also add a check if you want. Maybe you have a bean that is your favorite and you want to add it, and you can add it to the trial. So that's the first place. Then when you open and you click on a trial, you can choose a trait on the top. You have everywhere some little eye information in a box that gives you basic information on what it is as a traits. Same for the variety. As of today, and thanks to Sarah who gave me more information, I updated information for each of the varieties, so you will see more information now. And when you click on the little eye box, you can see more information now. On the 3 little dots, you can access the tracking date, the image, the comment, and report a failure. And you can have a direct access to the feed also right in here, as each trial have a feed like a Facebook group. And when you're done at the end, you can click complete. Everything is reversible, so you're really welcome to play, enter a review and delete it. Like it's not, we're trying to make the app user friendly, and so you really just try it. If you do a mistake, you can all the time reverse, delete, remove. So you can click in complete to see how it look like, and so forth. Talking about dates, right now, you can see 4 different types of dates. Planting date, the transplanting date is really optional for beans, so you don't have to enter anything. And then first and last harvest date. The first harvest is the date of dry pod for seed saving. That's when seed should rattle just a little bit, and that's the first harvest, and last harvest is when everything is ready. Within the next month or so, you might see another date, which will be the first flowering date. Again, everything is optional. Here, we really would love to have at least planting first and last harvest, and the flowering date. If you see the flowering date, we would love to have that. Key picture, that's going to become super crucial. So you can click on the 3 little dots in your app, click image, and we really love picture of like the apical meristem, like Sarah mentioned, on really to be able to give the architecture of the plant, the pod, the flower. You can have like how it is a type, if it's sprawling or if it's going up. So all those pictures are really interesting to have. You can tag each picture. That's as a new feature since this last summer. When you upload a picture, you can tag a trait. So maybe if it's the pod, or if it's flavor related to eating, or if it's disease or appearance, you can tag a trait if you want, and you don't have to. You have multiple ways to enter comments. You have a general comment box, and here would be great, all about the plant architecture and if it's bush or pole. You also have a comment box under traits. So if it's flavor, did you eat dry bean, or did you eat fresh green bean, or you can add different description of the flavor, or if you rate disease, you may be able to enter a disease name, and so forth. And so you add granularity to your review. You can also edit, like you can put multiple review for, let's say, disease. You may have early disease, late disease, and you can put multiple rating with some comments. And also you can edit and delete any review at any given time. Yeah, so some of the key traits, earliness, mentioned by Sarah, will be interesting to see how early. Flavor, height or structure can be interesting. Yeah, offline mode, is something that you can really use your web if you're in a place where you don't really have good reception. Just flip your phone, start from your house, open the app, make sure your trial is all loaded, flip your phone in airplane mode, and you can record in the field. And yeah, so when you're done with the trial, you complete, and then it will give you access to the live result as soon as you're done. You can see all the results. I can go over quick the communication. I don't know if some of you already use a feed. We have push notification on the phone. So like if Sarah and I would just do some posts, give you instruction, you will receive a notification straight on your phone. You can see those. You can, on your mobile phone, you can create a post, ask questions. Some of you already done it. And we can respond, we can tag you, and so forth. And that's really the main communication channel. It seems to work really well. We try to our best to respond. Yeah, so as feed creation, you can do a question, an update, you pick the trial you want. You can upload picture. You can even tag variety as ads, and so you can link variety, link people. Like here, tagging a variety name, tagging people. And yeah, when you're done, you have access to a lot of results from everyone on your desktop and on the app. And it's a very good way to look at everybody's results. You can look at picture from everyone, you can like them, you can see where they're from. On the mobile app, you can also see all the comments, as a picture. And you can create your own trial too. Outside of this trial, just you can create a wish list. From the wish list, you can track and create a trial straight from the app too, if you want to track other things. So that was a quick tour, and I can also, now we're going to open for question, and I can show the live platform for the Q&A. I'm gonna, okay. Let's
Okay, yeah, I just came back on so I could moderate the questions. We've been getting a lot of questions both in the chat and the Q&A, and Sarah typed in some of the answers, but just in case everybody hasn't been following that, we've been getting quite a number of questions about, just clarifying that the harvest date is for dry beans, but you're still encouraged to taste test and sample the green beans. Is that right, Sarah?
Absolutely. And the other question that folks wanted some clarification on was planting for unknown growth habit. So if it was a bush or determinant, we said, or if it's indeterminate, but a lot we don't know, plant as if it's a pole bean if we don't know. And you'll be pleasantly surprised if you have a bush and it's easy to handle, but expect a pole bean. Yeah.
That's
Okay.
great tip.
And the thing about cooking them, we don't know if these were bred for string beans or green beans, vegetables, or if they were bred for dry beans. But globally, most beans are consumed as dry beans. And so we assume they're dry beans. So we're asking you to collect that trait, and because we want to know when is the seed ready if we want to be able to share the seed. So please, like Alice said, collect dry bean harvest date, but feel free to eat them as green beans.
Okay, here's a question, I think it's for Nico, from Carol. It says, in the past, I've had additional varieties I would have trialed with the ones sent to me, but it gave odd responses, so I deleted mine. Has it been updated recently to allow more varieties per person?
So if your question is around the check and adding a check to your trial, you can only add one. And so we don't, within your own, within the trial, you can only add a check. If you create your own trial, we keep adding more and more variety. And if you don't see variety that you have, please send us a message at info or feedback at SeedLinked or Nico at SeedLinked. They all come to us and we can add varieties that you are not finding. We also have a form on SeedLinked where you can request adding variety, and we update those every month. So if it's in a hurry, just shoot me an email. I will edit them right away. Yeah, great question. Thank you.
Okay, great. Thank you. Let's see. Courtney has a question for Sarah. How far apart are you recommending planting each variety? The info says same growing conditions, so I was planning 3 rows, but may need to reconsider.
Oh, I think, okay, and that this will kind of answer Katie's next question. Grow the beans the way you would grow them as a home gardener. That's actually important for the trial. Is I grow them in ideal conditions in the greenhouse, and that's not the information that I want. I want how home growers would grow it in your area. 3 rows is fine. I'm assuming that's going to be on the same side of the house. I don't want you to do 1 row at your house, 1 row at your grandma's house, and 1 row at your uncle's house, kind of thing. Like the same 1/10 of an acre would be nice.
Yeah. Great. I see another question from, hey, Jay. Yeah, about green bean. My suggestion is you have a general comments. You can put information there. So the question was, would in the comment be a good place to record first and last date of green bean harvest? And if it's any good as green bean. Also in the flavor rating, you can also put a flavor rating and add a comment, green bean, and put a description there. I don't know what you think, Sarah.
I think that sounds great. I think, yeah, eating things I would put in there. Another trait that industry and that people are interested in beans is like canning quality and freezing. I don't know if any of your gardeners do that. Those comments, would they put them in the flavor or in the general comments section?
I would put in flavor, but also any general usage can also be put into general comment. And I will show you a little bit after how it is displayed. I see also a question about where to find information of each variety, so I can show all this maybe after.
So something that I think we're hoping to work out this summer is we didn't want to ask you to collect too much specific data at specific times, because that's no fun and that's not great. But as much data as you're willing to collect with the photos and the descriptions and the notes will give us an idea at the end of the summer of how much data we can expect to collect in the future. And so we'll organize it a little bit better. So we're happy to take constructive feedback on data collection.
Yeah, exactly. And especially how we design and SeedLinked evolve. SeedLinked is really built by you collaboratively. Like we take your feedback and we improve it, and it's a constant improvement. And so that's a typical project that is really made to see like how we can improve this. So that is.
Okay, we have a couple questions coming in about the growing conditions. So could you just describe the ideal growing conditions like light and water that we should be aiming for with these beans?
In general, common beans like full sun. Moist soil. I plant seeds 3 to 4 inches apart. Rows several feet apart. But it's different if you're growing them for a home garden or for commercial production. So something within that though, full light, moist soil. If they get a little dry in between, that's okay.
And you can find a trial management log, like I'm showing you here in the web, but you can find it on the mobile. Here in the little gearbox, trial management. And you can give an overall success of your trials. This is kind of a data quality index. If it was organic, field, backyard. And here you have a bunch of information about if it was irrigated, specific stress, and so forth. And that, which is interesting information, is that when you go into result and for like Sarah and other, and I'm going to take a trial result here. Let's see, of last year. And then when you look at all the results that you're going to be able to see, you're going to be able to play with those results, and maybe you want to look at only data that collecting on the drought, or you want to look at abnormal heat and so forth. And so then you can really look at data in many different ways. As I am showing here, I'm going to respond to a few questions on how to look for information on each variety. I just added more information today thanks to Sarah. So when you open here your trial of the bean trial, you have a little, and this is a web, but you have a little information here, and you can see I added more information, so you can see the origin, determinate, and so you have pod color, seed class, and obviously there's a lot of missing information, but this is where you can find it. You can also, if you go in the search here, in the search engine, you can look straight from your variety here, and you can pull the full card of each variety. And right now, you will not see much information, but as soon as people start filling information, you're going to start to see more picture, data, and so forth. So it might give you some tip of what other people are seeing, other comments. And so feel free to use a search to search the variety you have as well to see more information.
Okay, we had a question about whether it would be advisable to start the seeds indoors, given that there's such a small amount of each seed variety.
I would start them. I would direct seed them because I'm a survival of the fittest kind of gardener. They have relatively high vigor. We did a germ test right before we sent them out. If you're nervous, you're welcome to pre-plant and transplant, but beans don't really like being transplanted.
Okay, we had a question from Bridget here, who said she'll likely be growing her trial on certified organic ground. So it would be great to get a letter stating with a brief description of the trial, stating that the seed is untreated for certification purposes. Would she be able to get one of those if she requested it from you?
Yeah, please could you send this to SeedLinked to not bother too much Sarah, and we've done this in the past, so all the seed were untreated, so we can provide a letter, and your certifier will approve it without a problem. And as you know, for any trial and experimentation, you're allowed to have non-organic seed that are non-treated with a letter. So feel free to contact towards the end or whenever you need it.
Okay, great. Do you want irrigation information?
Yeah, you can, like I show on the trial logging, your trial log, you can, oops, there's too many things here. Oh, so here, trial management, you can, it's really basic, and so simplicity is what we need. So full irrigation, limited irrigation, dryland. You can also put basic command of your management here. We also ask organic matter and previous crop, and all those are optional. And so till, no till, minimum till. And I saw a question on how to review on the web. And so when you upload your trial, you just need to click on the trait and you can enter a trait here, a description, and save it. Here there are picture, here are comments, here is a date. Your planting date typically will be the same for everyone, so you could put like the date of now, and you can apply to all of those to not have to enter for efficiency. Again, here you see the description of each traits. Here you look at other traits. Here's how to complete. We also ask basic question: would you like to grow this variety again in the final process? And then yeah, and that's I hope that responds to the question.
Okay, let's see. Will the USDA be sequencing the genomes of the bean varieties? Cameron is wondering if there's a goal to begin associating alleles and the accessions with the gene bank to traits studied in the trials.
We would love to do that eventually. That is part of the strategic plan, but we're waiting on approved funding for that opportunity to happen.
And upon success of this pilot and validity of the information, as Sarah is going to grow them on her own as well, and is really going to give us insight, and I already have hinted that it's going to work really well, but really as a strategic initiative within SeedLinked is really to work on combining genomic and crowdsourcing data. Our advisor, you can see publication in Nature, who already validated the model. So if you want to get more nerdy, I can share some publication in PNAS and Nature. But it's coming. Yeah, it's a matter of a couple years, I think.
Okay.
Yes, blog about the trial, please.
Okay. Lucy's asking. She grows 90% in hoop houses. So, is that okay then to grow in a hoop house?
I think
with me.
Okay.
Is there a way to enter that in the SeedLinked data collection for the trial? Yeah.
Yeah. So you just record in your log. Yeah.
Okay, and Eric is asking, do you have a recommended date to cut off water for dry down, or recommended stage of the planting or something like that?
All these common beans should naturally senesce at the end of their growth cycle. And we don't know how many days to maturity any of these beans are. Once they start dropping a lot of leaves or forming a lot of pods, this is up to you. Again, just record how you grow them.
Okay, James is asking how to post to the feed on the app. Are there any tutorials somewhere, Nico, that could show how that's done for somebody that doesn't find it apparent?
Yeah, you're welcome to look at any tutorial on our YouTube channel. Let me go over it very quickly. So I don't know if you can see my screen here, but when you open your app, it's pretty much like X or any other social media. You have the little blue icon, you click, you can create a post. And when you click it, you can keep the privacy. It's public by default. You choose a trial, but if you are within the trial, the bean trial will be automatically selected. And then you can pick a question or update or recommendation. You can put a title and then type what you want, ask a question or update, upload a picture directly. I mean, it's very like any other social media. The difference here and very unique feature is that you can tag a variety. So if you use @ signal, you can tag a variety, somebody. So it's kind of really fun to look at that. And so let's see if I can go on the web. I can't really show you the app here, but you can see, and thanks for all of you. You've been very active on the feed already. Sorry, with Zoom usually. Oh, here we go. So if you're on the web here, on the general feed, you can click trial here. And then if you're part of multiple trial, you can pick, you probably only have one or two, but in my case, I have a lot. And so here I'm on the bean group here. You can see there's 213 people. And you can see there's already a lot of posts and question and excitement. You can follow each other. And from here, you can directly create a post and see it's automatically tagged by the trial here. Here's an update, but you can ask the question. Here you can, you can type a variety or people or.
Here's a question for Sarah. How were the 20 accessions selected out of the 17,000 possibilities?
Great question. So we distributed 210 seeds for this project, which is a lot more than we normally share. So first I chose accessions that had a lot of seeds that were verified pathogen-free, because we didn't want to be sharing bean common mosaic virus with anyone. And I think and then they had good germination. We did germination tests. So they're the seed there, and we had very little data on them, right? Things that are well characterized, we don't need characterized as much. And we wanted seeds from outside the US because that's a little more interesting. Maybe they are really going to be able to add some genetic diversity to breeding programs. So that's the whole purpose of this. We want to increase the genetic diversity in our food system and maybe regional diversity in those genetics. So that's how we selected it. We had seeds available, they're pathogen free, and they're from other countries.
Yeah,
Okay.
and as a question related to that, I've seen on the feed too, and I've seen here from Dina, some test date were like 1998, and it was like, as a grower, we like, oh, if we see a germ test from last year is the latest, as the oldest
Yeah.
we want to see, but here like 26 years.
Yeah, that's the difference between a seed company and a gene bank. That's why we don't give seeds to home gardeners. I did a rapid test the week before we sent them out to verify that they still had some germination, because yeah, some of the seeds are quite old.
Okay. Okay, are we to send the finished product back to you, or can we keep them to replant next year?
Please keep them and share them.
Yeah, and it's highly likely in the future we will pilot for you to save and send back to help curator to also, we will test if the quality and homogeneity is enough, and that would be amazing to see, and then that might be a really good way to help Sarah in the future. Yes, there's a great question on taste. What are the trade? Maybe that's Sarah. Yeah, bad taste. Yeah, when you taste green or at different stage, like maybe during in the feed, we can work with Sarah and we can do a post in the feed to give you a more elaborate response, but feel free to respond now too.
Yeah, and someone asked excuse me, an important question earlier about the toxicness of beans. So bean pods, you can eat them at any stage, but a bean seed has potential to contain lectins that can actually be quite poisonous. So once the seed is forming in the pod, you should cook them. You should no longer eat them raw. So for your stomach. Okay, then within that, yes, people know how to saute or eat the green bean raw, and that's delicious, or sauteed. For the dry bean, I soak them and boil them, and some of them taste good and some of them don't taste good. And I suppose we'll come up with more specific notes if people want, but I'm actually curious about your users, of how they evaluate when they eat a dry bean, if they enjoy it, if they enjoy the texture or the brothiness or the creaminess, or yeah, I'm new to that side.
And I will discuss with Sarah, but with SeedLinked, we have the capacity to create specific tasting trial, and then we have a much more granular type of traits, like texture, sweetness, bitterness, and so forth. And so maybe when we get closer to tasting, I might reach out to all of you and I can create some of those more specific flavor tests.
Oh,
So yeah,
that'd be
so
a lot of fun.
I might post some QR code in the feed that you can scan. And so stay tuned. Like I say, it's a pilot. And so I also really want to have suggestion, like, so go wild in the feed and give us feedback and how you want to do it. So.
Okay, great. Well, the questions keep coming in here. Joanna is asking, do we need to keep each variety isolated?
No, that's not necessary. Beans tend to self-pollinate. The male and female parts are right there in the flower together. By the time they open, they've usually fertilized themselves. There is a chance that insects will cross-pollinate them. So if you're saving your own seed, maybe less than 10% of them will be crosses.
Okay. Is it okay to blog about the trial?
Yes, please blog about it. Share it with all your friends.
Yeah.
Okay.
And then next year we're gonna have thousands of people.
Let's see. Are these bean varieties inbred or OP, or are any of them hybrid?
None of them are hybrid. They're all inbred. They're all inbred or OP. They've been grown in a greenhouse, and we don't control. There's no insects in the greenhouses.
Okay, here's a good question from Jim. Why do some varieties have numbers instead of names?
Great question. We just don't have enough information on these. Sometimes varieties have numbers if they're not released, if they're part of a breeding program. Sometimes varieties get released with the number because they have bad marketing. If it has a name, it's likely a variety that, or a landrace that somebody cherished enough to give a name to.
If there's a code, can people suggest a name or no, it's impossible to change?
If it ends up being everyone's favorite variety this summer, I'm sure your growers could give it a name and some seed company will pick it up and make those available. And that would be a success story for year one.
Okay, come up with some name.
Yeah.
Yeah, I was reading a short story not long ago about somebody who loved the pumpkins in his garden so much that he gave each one of them a name.
Yeah.
But okay, so let's have there are a couple more questions here. I'm guessing you'd like to have us taste test the beans both as snapped and as dry beans. The ones that are not palatable as a green bean may be easy to tell as they have a woody or bad taste, but what are the traits that would suggest that they are not good as a dry type, or are all beans good as dry beans?
They're all edible as dry beans. Some take longer to cook, some have thick skin. A lot of it is personal preference, but there is a range of flavors in dry beans. Some are more nutty or buttery or vegetable-y or starchy. They get pretty complex profiles, and they also have different textures as they dissolve into the broth. So for different recipes, you'd want them for like a bean salad where they hold together really well and stay crunchy, or you would want them for what do you call, like a hummus type dish.
Or cassoulet. I'm French, so you will see. There's one of the bean, if you have Alaric, is the famous French cassoulet bean I added to the trial. And it's amazing, yeah, the difference of texture, creaminess, buttery, skin thickness. Yeah, I think huge in texture. Like we'll try to bring a chef and we can do a post in the feed to talk about some of those. Oh yeah.
Oh, fun.
Okay, great. Let's see, we have another question or two. Which seed bank did these come from? These came from all over the world, right? Or
They originated from all over the world, but we keep them here in Pullman, Washington at the W6 is what we call ourselves, or the Western, we have a really long acronym. Plant introduction.
oh, okay. Michelle asked, if we live nearby, can we come visit the lab?
I assume you're talking about the gene bank. And yes, we do give tours, a lot for like student groups or master gardener groups or something. But you can reach out and we can organize something. We like to show off what we're doing.
Okay, let's go Cougs from Moscow. Okay, I think we've gotten to the end here. But what should people do, Nico, if they have additional questions?
yeah, please post all your questions in the feed. I know some of you don't like so much media and mobile phone and so forth, but this is really for a good cause, and so it's not Instagram or Facebook. And so I would really highly encourage you to post everything in the feed of SeedLinked via the mobile app or via the web, and you will share to the whole group. It will be easy for everyone. You can tag, and so yeah, thank you. And again, thanks everyone to join and to be part of this pilot. I'm super excited to start this project with Sarah and USDA, and we tested the model with Seed Saver and Adapt, and so it's really a collaborative effort to get to where we are now, and it's not going to be perfect, but you will be surprised at the end by the vast amount of information, and how really in the future the dream is all this information to be aggregated as a search engine for scientists and breeder to be able to look for a bean and use data to start locally adapted French cassoulet bean for, I don't know, for Corvallis or
Sounds good.
So thanks everyone.
Thank you, everyone. I'm so excited by your participation and enthusiasm and the data collection. Yeah, I think we're going to be successful this year, and it will be the first of many years. And so we're starting with beans, and I think Nico put it in the letter. Beans are exciting because they're good for the soil, and they're good for your health, and they're good for the environment, right? But they're also delicious. And I see people posting that they have fancy chef friends who are excited about beans, and that's what we need to do, because they're not just poor people food. They are this sexy future of
there we
food
go.
systems. So thank you guys for working on that.
Yeah, and we're highly likely going to feature this in the variety showcase of Lane Selman in September in Portland. If you're around, is amazing events, and so I'm sure some of them will be featured there. And I want to finish. Thanks so much, Alice and eOrganic, to help me host this. I will not do it on my own. And Alice has been amazingly volunteering to help me and hosting this wonderful webinar. So thank you so much.
My pleasure. Okay, bye everyone.
Thanks everyone. Thanks, Sarah.
Thank you. Take care.
Bye.